Ancient mating patterns shaped the human genome. New research from the University of Pennsylvania reveals that ancient mating preferences, not just biological incompatibility, played a significant role in shaping the modern human genome. The study confirms that prehistoric humans and Neanderthals frequently interbred, but not randomly. Instead, there was a consistent bias towards Neanderthal males mating with modern human females. This pattern explains why Neanderthal DNA is almost entirely absent from the human X chromosome and highlights the surprising influence of social behavior on our genetic history. The Neanderthal deserts and the missing DNA. For years, scientists puzzled over Neanderthal deserts, large stretches of missing Neanderthal DNA on the human X chromosome. The initial assumption was that these gaps occurred because certain Neanderthal genes were harmful to humans and natural selection eliminated them. However, this new analysis suggests a different explanation. Researchers re-examined Neanderthal and modern human genomes and found a striking pattern. Gene flow occurred predominantly from Neanderthal males to modern human females. Sex bias in interbreeding. The study's key finding is that Neanderthal males partnered more frequently with modern human females than vice versa. This bias explains why the human X chromosome contains so little Neanderthal DNA. Because females have two X chromosomes, while males have only one, the direction of mating significantly impacts which genes are passed on. The research team identified modern human DNA in 3 Neanderthal genomes, comparing them to a control group of African populations that never encountered Neanderthals. The results were clear. Neanderthals had an excess of modern human DNA on their X chromosomes, while modern humans had almost none on theirs. Why this matters? This discovery is crucial because it challenges the traditional view of human evolution as solely driven by survival of the fittest. Social interactions, specifically, mating preferences, were a powerful force in shaping our genetic makeup. Roughly 600,000 years ago, when modern humans and Neanderthals diverged, populations migrated and interbred. The study demonstrates that these encounters weren't random. They followed distinct social patterns. The researchers ruled out reproductive incompatibility or toxic gene interactions as the primary barrier, leaving sex-biased interbreeding as the most likely explanation. Modeling the results, mathematical models confirmed that this mating bias could reproduce the observed genetic patterns, while other factors, such as sex-biased migration, could theoretically create similar results. They would require far more complex and inconsistent scenarios. The simplest and most direct explanation remains that Neanderthal males were more often the partners of modern human females. Mating preferences provided the simplest explanation, said Dr. Alexander Platt, the study's lead author. The findings appear in the journal Science, suggesting a fundamental shift in our understanding of human evolutionary history. In conclusion, this research underscores the complex interplay between biology and social behavior in shaping our species. The story of human evolution isn't just about survival. It's also about who our ancestors chose to mate with and how those choices reverberate through our genes today.